All right, so here we are with an overview of biochemistry. So this, all this information came from this YouTube channel named Professor Dave Explains. So I feel like I got pretty lucky. Usually I shop around and try to find the best type of material, and it's usually kind of hit and miss, and I have to shop around for a while. But I got lucky this time. This guy's pretty good. This is the link to the beginning of this playlist called Biochemistry. And you can also just look for Professor Dave Explains Biochemistry, and this playlist will come up. So let's get started with word of the day. Now, um, medical term, medical terminology of the day. So, the word of the day is neurite. Any of the branch processes extending from the cell body of a neuron. Neurite. Okay, pretty straightforward. Neurons, neurite. So, moving on to the overview of biochemistry. So, essentially, he covered all of the four major biomolecules: proteins, carbohydrates, lipids, and N acids, aka nucleic acids. So what I hope to do with this is, I consider his overview of biochemistry in that playlist, I consider it to be a very good summary of stuff covered in at the undergrad level. So moving forward, the stuff, these notes, believe they can be used as a scaffold uh, from which to um, on the which to add all of the details from the upcoming medical biochemistry course. So we'll start out simple, and we'll have this simplified information, but then slowly find out where any given topic. So let's say, PDF file number 3, and it has some topic about lipids. So we would link all of that information back to lipids, and then there might be some information related to lipid proteins. So I would proceed to draw arrows and create a mind map to the original notes, just to see how at the end, by the time of the midterm, how everything interrelates is interrelated to each other, and how things come together. So without further ado, let's move on to the beginning of the information regarding proteins. So it's pretty straightforward. You have this convention of putting the nitrogen first, and then a carbon and another carbon. And the carbon here in the center, this is the alpha carbon. And moving forward, a lot of the hydrogens will be implied. It'll be assumed that we all have a good handle on where the hydrogens are. And if there's a double bond, for example, someplace, you can expect to also have the hydrogens implied, just remembering how many there are supposed to be. And it really makes things neater. There's much less clutter if we leave out hydrogens in a lot of these notes. So again, at the very foundation of proteins, you have your NCC. And I think of it kind of like CNN, the news channel, except backwards. Instead of having two N's, CNN, you have two C's and a single N, so NCC. And this center carbon is the alpha carbon. And that one obviously has an H there, but moving forward, I won't include that. So coming off of the alpha carbon, that's where you have your R group, aka your side chain. So let's move on over here. This is another way of drawing. This will be the way that I draw the proteins from now on. So again, this is the green will from now on will symbolize nitrogen, and then the black will symbolize carbon, and here's another carbon. So again, NCC, and the R group in this case is this magenta or bright pink. And finally here I've included in the, I believe it's the beta carbon, I have included these two oxygens, which are written drawn in blue. I have included these oxygens in order to exemplify the fact that this is the C terminus, and that's where this carboxylic acid carboxylic group is located. And where do we go next? Okay, so another important topic that that Professor Dave covered is in the playlist is that, you know, here you have a any given um, amino acid with both a plus charge and a minus charge whenever the pH is neutral. So the center means neutral pH. Whenever the this molecule, this amino acid finds itself in an acidic environment, it will easily get protonated, and you'll have a plus charge over here on the nitrogen side on the N terminus, and you'll have a neutral charge over here because this hydrogen will be um, put on this hydrogen will be put on this oxygen. Yeah. So, and then over on the opposite end, whenever the amino acid is in a basic environment, it'll have a minus charge over here and a zero charge, a neutral charge on the nitrogen because it has just the right amount of hydrogens to make it neutral. But then this will be deprotonated, so there will be a minus charge on this oxygen. And again, a lot of things are just implied. So here, right here in this area, you would know that there's a lone, maybe, is there a lone pair of electrons up here? Maybe. It doesn't seem like that's too critical for the purpose of this class. So if necessary, we'll review that. But for the moment, based on what I've seen on the PDF files, those notes provided by the professors, based on what I've seen, it doesn't seem like we need to go back to all of the tiny details regarding all of the biochemistry, all of the actual chemistry. So we'll be okay moving forward. So another bit of good summary here is that under acidic conditions, there will be on the amino acid, not including the R group, excluding that, just considering the NCC backbone of the amino acid, you'll have 5 hydrogens total, 1, 2, 3, 4, 5. And then under neutral conditions, you'll have 4 H total. And then under basic conditions, you'll have 3 hydrogens total. And again, those are cations and anions. So moving on. Okay, so again, this is located directly below, which means that we are working with this with this amino acid in neutral conditions. So you have your plus there, and you have your minus. All right, let's move on. So this same amino acid is copied here 5 times in order to highlight or exemplify or show what is what is being shown, so to show that how the polymerization is going to take place. So here in this yellow box, I've highlighted this oxygen atom. Sorry, yeah, this oxygen. And then those two, and a lot of this will be conceptual. This is in the actual chemistry. This is just for the sake of highlighting what's going on. So conceptually, that oxygen and that hydrogen can become dissociated from this amino acid and from the adjacent amino acid. One of the H's, one of the hydrogens on the nitrogen gets gets dissociated. So what do you get when you have an oxygen and two hydrogens? You get H2O, aka water. Hence, this is a dehydration reaction in order to create the peptide bonds. These are dehydration reactions, and um, that's how you get your polymer. This is how you get your polypeptide. So after making so this is this dark yellow bond. It's written in this color instead of gray, just to symbolize that this is the new bond that was created. And specifically, it's a peptide bond. So here you have um, your polymer, your polypeptide, but just condensed so that it can be visualized better. This is the exact same thing as above, but with the bond a little bit shorter. And remember, you're going to have your N terminal and your C terminal. And here's the definition. An amide is a functional group with one nitrogen atom next to a carbonyl. There you go. So that is all of the information that I thought was relevant for this course. Now all the information relevant to proteins. So now let's move on to the next thing, carbohydrates. So there's a number, let's consider the number of carbohydrates, the number of carbons that a carbohydrate can have. So when it comes to one carbon, it doesn't exist. There's no sugars, no saccharides with a single carbon. Likewise, two carbon sugars do not exist. So we're going to ignore that. But here, with three carbons, that does exist. And when it comes to red bonds, those will be done whenever there's double bonds, and it's going. The red double bonds symbolize carbonyl, symbolize the carbonyl. So when it comes to three carbon sugars, there's always two things to consider. Where is the carbonyl located? Is it located at the extreme, at one of the ends of the molecule, or is it located somewhere in the middle? If it's located at one of the extremes, one of the tips, that's called an aldehyde. And if it's located somewhere in the middle, somewhere that's not at the tips, that is called a ketone. And here, let's fix this. This just got messed up a little bit, but there we go. Glyceraldehyde. So something else, an important thing that Professor Dave had in his playlist is that there's a way to name these molecules. So if a sugar has three carbons, it's called a triose. If a sugar has four carbons, it's called a tetrose. 5 carbons, it's a pentose, and then 6 carbons, it's a hexose. So, furthermore, if any given sugar is an aldehyde, it's an you'll name it accordingly also. So when it comes to this 3 carbon sugar, it's a triose. but specifically it's an aldotriose. And then when it comes to this this other 3-carbon sugar, it's a ketone, so it's a ketotriose. Ketotriose. So let's see. Um, so before even considering a sugar, let's backtrack and just consider this simple molecule called glycerol. And what do we have? What is it composed of? A carbon, another carbon, and a third one, and with some hydroxyl groups. And again, here the H's would be implied. Let's just draw it like this for simplicity. So you have this as the foundation for a lot of things. So if you take a glycerol, but then and then you add a carbonyl group to it, that's when you start getting into the realm of sugars. So the name here, as you might expect, or as you might remember, is glyceraldehyde, in reference to glycerol. We started out with the glycerol essentially, but now this is a sugar, glyceraldehyde, and here you go. Here's the aldo. The ALD represents the aldehyde character of this molecule right here. Cool. So, and whenever the whenever the carbonyl is not located somewhere at the tips, then that means that it's a ketone. And since this molecule, since this sugar has only 3 carbon molecules, then it only has one option of where to have the carbonyl away from the extremes, and it's right here in this second carbon. And then that also can get you started on a conversation about stereoisomers and diastomers. That's also something that you may review if you feel like it's necessary, but I haven't determined if it's necessary based on, well, for the context of this course. Um, so similar to how we referenced glycerol over here, this simple molecule, just all carbons with some hydroxyl groups. Likewise, we can reference another molecule over here. So when it comes to this acetone, it's 3 carbons with a carbonyl, but there are no hydroxyl groups elsewhere. So what happens when you start modifying this starter molecule? Well, you get dihydroxyacetone, and it's called dihydroxy because you started out with just acetone right there, but then you added di, aka 2 hydroxyl groups, 1 and 2. Alrighty, so all this is making sense coming back to me. I hope you are having a similar experience. So, FYI, whenever there is this purple asterisk, it represents a chiral center. So, and here is a review. You have the option of reviewing Fisher projections. So in case that becomes relevant within the course. But for now, let's move forward. So we've covered a lot of information regarding sugars with 3 carbons. So let's move on to sugars with 4 carbons. Cool. Here we go. Not much to it. Just, keep in mind what is the location of the carbonyl, and that makes it and if it's at the tips, that makes it an aldehyde. If it's anywhere in the away from the tips or the extremes, that makes it a ketone. And in this case, there are 2 chiral centers right here, and over here there's only 1 chiral center, based on just based on the nature of how the ketone is formed, based on the nature of where the carbonyl group ends up and the limitations of the number of carbons. So moving forward to sugars with 5 carbons, and right here this is highlighted in orange right now, and there's some arrows that lead away that'll become relevant later. For now, let's just focus on the number of carbons. There you go. You got a 5 carbon aldehyde, otherwise known as an aldopentose. Yep, aldopentose. And down here you have a ketopentose. Cool. So not much information there. What do we have over here? Okay, this is a link to review what it means to make a ring for a sugar to make a ring. So essentially, an alcohol, where one of the hydroxyl groups attacks a carbonyl, and then based on the size of the molecule, certain um, certain hydroxyl groups on certain carbons will attack the corresponding carbonyl, and you'll get different results. And here's something important. When it comes to the concept of an anomeric carbon, this is the carbon with the carbonyl which was recently attacked. And moving forward, so not much information here, or at least not yet. We'll come back to that. So now take into consideration this sugar with 6 carbons. Oops. We'll be focusing on the aldohexose. For now, let's ignore the ketohexose. So we'll take this aldohexose and move over here and see what happens whenever a hydroxyl group begins to attack a carbonyl. So here in orange, you'll see 2 options. This right here and this right here is the same molecule, except that here the oxygen on carbon number 4. So, and something else to note, whenever there's something special about a carbon, for the illustrative purposes, I've swapped out the color from black to brown, just because it'll be easier to see, easier to keep track of that carbon later down the road. So in orange here, and also I've changed the color of the oxygen from blue to purple, also to keep better track of what's going on with it. So in this case, it's carbon number 1, 2, 3, 4. The carbon number 4 that has a hydroxyl group attacking the carbonyl. And in this case, it's 1, 2, 3, 4, 5, this hydroxyl group that is performing the attack. So what happens in each case? Well, just look straight down. These are the ones to consider together. So if the hydroxyl group on carbon 4 is the one that attacks, you get something called a furanose, and you get this pentagon, pentagon ring, and you get here, so you get the carbons in the corresponding place. So remember, this carbon was the number 1 carbon in brown, so here it is again. And the bond that was created between the attacking oxygen and the carbonyl group, I've drawn it similarly in orange, so we know which is the new bond that was created. So here you go, and then here is the rest of the molecule. So this is carbon 1, 2, 3, 4, 5, and 6. Cool. Alrighty. So right next door, whenever the hydroxyl group on carbon 5 performs the attack, you get this thing called pyranose, and you get the hexagon shaped ring. So moving on to an example. So something that's really famous is glucose. And this is what glucose looks like. And excuse me, it's worth reiterating that glucose is a pyranose. Ta-da! Pyranose. So in this case, I've numbered the carbons for illustrative purposes. So this molecule that you see here, I've just copied it 3 times in order to exemplify what's going to happen next in terms of polymerization. So here you have this hydroxyl group on carbon number 1, 2, 3, 4. 4, this hydroxyl group on carbon 4 is going to perform some sort of chemistry on carbon number 1 of this adjacent glucose molecule. Hence, that's why you get the concept of 1,4 glycosidic linkages, because these kind of these specific hydroxyl groups are performing chemistry chemistry actions on corresponding carbons. So the end result is this glycosidic linkage. So this blue oxygen that you see here, I've conceptually just moved it to the center in order to see what was happening. Again, this brown carbon is here just to keep track of the fact that this is carbon number 1. And then the same thing will happen over here. Whatever happened for this linkage, it can also happen over here. But for saving time, we won't get into that too much unless it becomes necessary during the course. So here are some famous names. So you got cellubiose, which is a beta glucose and another beta glucose. Oh, and that's another thing. So remember these hydroxyl groups right here. These can be either up or down, and those will represent different types of molecules, beta glucose or alpha glucose. We'll get back to that if necessary. For now, whatever. We'll just take it as it comes. So again, let's go back to famous names. Now it's a good time to, let's see, add a little bit of flair to make this more memorable and a little bit more enjoyable. So Hollywood Walk of Fame. There we go. That's a good picture. Yeah, that's a good picture right there. Okay, cool. So famous names. Here we go. Hollywood Walk of Fame. So cellobiose, that's what that's made of. Lactose. These are the two monomers, monosaccharides, which that's made of. Sucrose. There you go. And there you go. That's the combination. So when it comes to starch, starch is made of many alpha glucoses put together, and they are put together with 1,4 glycosidic bonds. When it comes to glycogen, there's it's the same thing as starch, except that there's so much more branching. And when it comes to cellulose, it has many linkages, 1,4 glycosidic linkages between many beta glucose molecules. Cool. All right, so that's about it for famous names, and that covers everything related to um, carbohydrates. Anything that seems important, at least for now. This will get us going. This will be a good way to start. So now we can move on to, let's go back and review the list. It's pretty straightforward, but again, it's prot. That's my shorthand notation for protein. You got carbs for carbohydrates. Next, we'll cover lipids and then nucleic acids. So here we go. Lipids. So this is an important bit of review. What's the difference between a carbohydrate and hydrocarbons? Well, here we need to make a change here. Cool. So when it comes to carbohydrates, as Professor Dave explains in his video, it's carbon that is that's hydrated, hydrates of carbon. So those carbohydrates, they include the following elements: carbon, hydrogen, and oxygen. And when it comes to hydrocarbons, the only two elements which are involved are carbon and hydrogen. Oxygen is out of the equation. As you can see, that's why it's crossed out and written in light gray, because we're not taking that into consideration when it comes to hydrocarbons. All right, so some major characteristics of lipids are as follows. Lipids are not polar, aka um, not polar, yeah, just not polar or apolar. They are made of long hydrocarbon chains, or they are made of ring systems. Some examples of lipids are fats, oils, or steroids. So, um, here are some concepts that may become relevant later: ester groups and acyl groups. So we'll see if that's necessary. So, all right, so it's important to go back and examine this glycerol molecule. So where is this green arrow coming from? So here we go. So yeah, earlier we were talking about the concept of this starter molecule. Where do we start? Well, we start with the glycerol, and we use that concept of the glycerol to start talking about sugars. There you go. And likewise, the reason this is highlighted is because we're going to consider the concept of glycerol, and we're going to follow the green arrow down to where it's relevant again when it comes to lipids. So again, this is just a glycerol molecule, but then it's going to combine with um, individual fatty acids. I call them F acids for short. So that those kind of interactions are going to happen. So here you have again the glycerol molecule. It's the same molecule, but the hydroxyl groups, I just pointed them down. And here you see the glycerol molecule interacting with three individual fatty acids. And that's how you get a lipid, or one type of lipid. And this is before it has the polar head group over here, which is what the lipid bilayer is made of. So for now, just let's just appreciate this concept right here, which is called TAG. And TAG stands for triacylglycerides. These are tags. And this right here, that's surrounded by this pink box. So anything that involves all these carbons and all these hydrogens and this one carbonyl group, that's something called the acyl group. So hence the name triacylglyceride. Glycerol right here, and then tri, you have three acyl groups. These are tags. There you go. So what else is going on? So uh, in this case, we're still examining a single individual fatty acid, but just in isolation, to examine its various, examining the anatomy of the fatty acid. So here you go. Here you have the carboxyl group. That would be the equivalent of CO2. as after chemistry has been done in order to make it mingle with this hydrocarbon tail. So as you can see, we go back to this important review of concepts or terms. When it comes to a hydrocarbon, it involves only carbon elements, the element of carbon and the element of hydrogen. So hence why here the hydrocarbon tail is only this, only whatever is surrounded by the green, and notably all there is carbons, and I didn't draw the hydrogens, but they are implied. So what's next? Here we go. So as I mentioned earlier, this is a tag, triacylglyceride, and the tags are used for energy storage. That's definitely important for the class, for the upcoming weeks and months. So here you have your tag. The next question to ask is, yes or no, does the hydrocarbon tail contain the maximum number of hydrogens possible? If the answer to this question is yes, then this fatty acid is saturated. Yes, it is saturated. But if the answer is no, then this is a not saturated fatty acid. In other words, it's an unsaturated fatty acid. So in naturally occurring fats, double bonds are almost always cis. Trans fats seem to be harmful to the body because their linear shape creates a higher melting point. Trans fats are more likely to remain solid, so they become difficult to metabolize, to be metabolized. To metabolize. Okay, there we go. The correction is done. And there's some more concepts to cover here based on the Professor Dave playlist. For example, what does it mean to be polyunsaturated as opposed to just regular unsaturated? We'll cover that later if necessary. There's also the concept of omega fatty acids, and that has to do with where there exists a double bond, and all of that we'll cover later if it becomes relevant. Now, based on this definition up here, we said that lipids can be made of long hydrocarbon tails. Check, we did that with tags, but they can also be made of ring systems. So let's see what that entails. So a subtype of lipid or a subcategory of lipids is steroids. And as Professor Dave mentioned in the playlist, steroids don't include just anabolic steroids, which are used for bulking up, getting more muscle. It also involves a wide variety of other molecules. In particular, it involves hormones, which are obviously very essential in for everyday living, for your health on a daily basis. So we'll be dealing with ring structures like that, like this hexagon, and like this pentagon. So let's move on. Ultimately, this is what all steroids look like. This is the building blocks for them. You have these four rings, and they are labeled as follows: A, B, C, and D. Furthermore, at these vertices, you add a carbon. And again, the carbon here, I drew it in brown because it's unique. It's not part of the ring, and it's just a lot easier to see. So here, that's why you have this brown carbon. What else is important about this? Well, you have to know the specific way to number this ring system. So always think of this carbon right here, just memorize it, and this is carbon number 1, and then memorize this carbon right here, it's carbon number 11. So having put this into rote memory, we can move forward to more details. So when it comes to carbon number 1, you're going to start numbering in this way. You go around in a counterclockwise fashion. So this will be number 2, number 3, number 4, etc., etc. And I'll illustrate that coming up. And when it comes to 11, this one's already numbered, that one's already accounted for also. So in this case, you follow this route. You start out from 11, you hit that one, then this one, come around, da-da-da, and then once you reach the last one, then you jump onto this special brown carbon, and eventually you also go over here to this other special brown carbon. So ultimately, once we've done all the numbering, this is what it looks like. Once you follow the conventional route of numbering, going around the ring system in a specific way, this is the numbering that you get ultimately. And then at the end, once you reach 17, that's when you jump onto the special brown carbon, and you got 18, you got 19 carbons total. And again, a lot of things, all of the hormones coming up for both genders will be based on this structure. So let's move on to examining something special that happens at carbon number 17. Here I didn't label it, but remember, this one is 17. This topmost within the ring system, number 17 is this carbon up here that is the topmost, furthest to the right, but it's the topmost. So 17 is special because a lot of here we go. Carbon 17 is a common place for alkyl substituents. So lots of things are going to get attached at carbon 17. XYZ just stands for fill in the blank. Whatever type of hormone you're trying to build, this is where a lot of the action is going to take place. So furthermore, cholesterol seems to be a very important type of lipid. So here I've highlighted what cholesterol looks like. It has this substitute up here on carbon 17, and these are all carbons, but I drew them in purple because they're special carbons. They are what compose cholesterol, and that's so critical for biochemistry. So there's 1, 2, 3, 4, 5, 6, 7, 8 additional carbons that get substituted or added onto carbon 17. And again, this is definitely more conceptual than conventional. This doesn't adhere very well to Hayworth projections or Fisher projections. This is for illustrative purposes. Hopefully this is another perspective that may provide assistance for you to make sense of this class. During the Professor Dave lectures in his YouTube playlist, he also covered the various types of hormones that exist in males and in females. But in this case, I won't get into that. We'll get to it if necessary, but for now, just know that you'll definitely stick with this. We'll be sticking with this main ring system that's kind of the foundation for all those other hormones that they include estrogen and the testosterone and progesterone. So we'll cover that if it becomes necessary, which is why I left a lot of space here. This is where those notes would go. So that covers everything related to lipids for now, everything that seems important. So let's move on to N acids, aka nucleic acids. So let's go back and follow this orange arrow. This is important because, we will be referencing this aldo pentose. How is it important, this 5-carbon sugar? Let's find out. Or you may already know, depending on how much of review this is for you. So moving down, we're following the orange arrows, and here we go. We have arrived. So that aldo pentose is important because it is how ribose gets synthesized. So here is this illustration from this website, chem.libretext.org, and it just shows you that it is the hydroxyl on carbon number 1, 2, 3, 4. There you go. On carbon number 4, that hydroxyl performs chemistry action on the carbon number 1. Ta-da! There you go. And you get this pentagon-shaped ring. That's ribose. And I drew it myself here based on the colors that we've been using before. And remember, this is an oxygen, hence the letter O, but it's drawn in purple because that is the hydroxyl group right here that used to be on carbon 4, but still here on carbon 4. But now it has created this bond with carbon number 1. And again, this is in brown, light brown. And the bond is in orange because it's a special bond that was created recently with that creation of the ring. And again, remember, this is the anomeric carbon. Cool. So let's move on. So you've got your ribose, which is a pentose. So how does that incorporate into the concept of nucleic acids? Well, here is the pentose, which we were working with. There you go. Now, chemistry happened, and they got attached to this thing, which is the base. And again, it's called a base because right here there are there's electrons, I believe. So this is basic. So that's why it's called a base. And then over here you have a phosphate group. Moving forward, the phosphate group will be always drawn like this. The P for phosphate, I'll draw it in red just for it to stand out. And there will be always 3 oxygens in blue surrounding it. And it's worth mentioning that, yes, I know that one of these has a double bond. Let's say the top one, 1, 2, and then the rest have single bonds, at least when they're drawn based on convention. But for simplicity's sake, I just drew the oxygens in close proximity in association with the P. So that's just for illustrative purposes. So this whole thing is a phosphate group, and there are specific bonds implied, but we'll cover that later if it becomes critical to understand those specific details. So for now, this is what we have. This whole thing is a nucleotide as compared to a nucleoside. So what is a nucleoside? Well, here is the definition of a nucleoside. It's everything. So this whole thing is the exact same drawing that was over there before we came over here, except that here in this pink rhombus or diamond, I have enclosed everything except the phosphate group. We've excluded that. So this molecule without the phosphate group, that is the nucleoside. So only the side, nucleoside, as compared to nucleotide. Everything is tied together. All of the three components, the base, the sugar, and the phosphate. So that is the nucleotide all tied together. Okay, so this drawing here, so whatever you see here, all of that is drawn again down here. And it's just copied and pasted for the purpose of asking this question. So here at carbon number 2, again, that's 1, 2, 3, 4, 5. The brown is special because that's the anomeric carbon. So at carbon number 2, ask yourself this question. At carbon number 2, does there exist a hydroxyl group? If the answer is yes, then this molecule is a ribose. Then this is, it'll go on to create, just to be a component of RNA. If there is a lack of hydroxyl group, then this is deoxyribose, meaning, you know, this word or this prefix the, it means that the ribose sugar has been dehydroxylated, hence the name deoxy. So there's only one hydroxyl group here at number 3. Here, this one we would just erase it, and boom, that's how you get deoxy deoxyribose, deoxyribose, and then that will later create DNA. Cool. Okay, pretty straightforward. So then we have this arrow pointing to this diagram that I got from this website. If you'd like to go check it out on your own. And it's just this undergrad level comparison of DNA versus RNA. Here you go. And then over here, these are screenshots from the Professor Dave Explains playlist. These are the DNA bases. And just a thing to note is T versus U, and T would have a what's that called? Methyl group at this vertex, but it doesn't. So that makes it uracil, the letter U. All right. And over here, I've illustrated how a DNA molecule and either an RNA, in this case it would be RNA because you have 2 hydroxyl groups right there. Here I've highlighted how a nucleic acid would be made. So you have again 1, 2, and 3. So this is carbon number 3. The hydroxyl group from there will perform chemistry action and create a link to the phosphate of the adjacent nucleotide. And again, it's highlighted here. This third hydroxyl group, on carbon 3, that interacts with the phosphate group below, and that's how you start to form the phosphate backbone. So if you follow that, it says there are bonds all through here, hence the name phosphate backbone. And I only drew 4 nucleotides here, but that should be pretty self-explanatory. These 4 can extend downward to 8, 16, and upwards to several millions. Hence why sequencing can take a long time. There's a lot of nucleotides. So hopefully, I'm pretty sure all of that is all very straightforward review. So the next important thing to point out about nucleotides, you know, we're still within this topic of nucleic acids. These will be relevant later. You have ATP, which is again this nucleotide, and typically the nucleotide would just be would have a single phosphate, but as you probably remember, with ATP, which is used for energy purposes, you have three phosphate groups, hence the tri right here, hence the letter T, triphosphate. So some important notes regarding the ATP is that yes, these phosphates they're eager to come apart. How do they come apart? Well, hydrolysis of ATP happens, and that's when they fall apart. So more details later, if necessary, for hydrolysis of ATP. So all around these phosphates, all around this area, there exists an excess of electron density. So that means that there's a lot of potential energy stored here, similar to a spring that is compressed. Just wait, all of that potential energy is ready to be converted into kinetic energy. Something else important to point out about nucleic acids is the existence of cyclic AMP. So what happens is that one of the oxygens from the phosphate group, one of those oxygens reaches down to the hydroxyl group, or well, one way or another, there's a bond that happens between this hydroxyl group and this oxygen of the phosphate group. So instead of creating a bond between itself and another nucleotide, it creates a bond with itself, hence the name cyclic AMP. So that's pretty important also, and it'll be relevant later. Finally, here we are at what will definitely be important during this course: metabolism, metabolism, metabolism. All right, let's take a minute to do a little breathing exercise. Take a little break. I need a charger. Breathing exercise.
This is a deep breathing exercise that will allow your head 7, if you aren't exact here, 2, 7,
Okay.
as you gently close your eyes. You'll inhale through your nose for a count of 4, hold the breath for a count of 7, and exhale through your mouth for a count of 8. Don't worry if you aren't exact here, just do your best. Inhale 1, 2, 3, 4. Hold 2, 3, 4, 5, 6, 7, and exhale 2, 3, 4, 5, 6, 7, 8.
All right. Yeah, that's pretty helpful. So let's jump into metabolism. So again, here is again ATP, just drawn here for illustrative purposes. And here's a summary of the three types of, or the three concepts involved in metabolism. You have G-cis, aka glycolysis, and then you have the citric acid cycle, and then you have oxidative phosphorylation. So here's a summary of things. I'll fill this out later. I just filled out the top. Where does glycolysis take place? In the cytoplasm. It does not need oxygen. There are 10 enzymes involved, etc., etc., etc. And in this case, it would be helpful to go ahead and jump to the bottom, and let's copy and paste this. So copy, let's go down to the bottom, because in the playlist, Professor Dave provides this summary of all of the three concepts. So in this case, it's a good idea to jump straight into a summary to give us an overview of what's happening in regard to metabolism. So as noted here, okay, so this is a mitochondrion. This is what that represents. So anything outside of it, obviously, is cytoplasm, and here's where glycolysis is taking place. Cool. But then within the matrix, as noted here, the citric acid cycle is taking place within the mitochondria matrix. So that's why the Krebs cycle is taking place here within the mitochondrion. But then oxidative phosphorylation is taking place in the mitochondrial inner membrane. Hence why the illustration zooms into this lipid bilayer of the mitochondrion. And here are some details. All right, very good. So now let's go back up to the top so we can start to look at some of these details. So here we are. Here we are. Metabolism, metabolism, metabolism. Cool. So whether we are consuming starch or whether we are breaking down Ggen, aka glycogen, from storage in the body, what matters is that one way or another we'll get glucose. And then glucose will have chemistry done on it and it will provide energy. Something important to point out is this molecule, NAD, which stands for nicotinamide adenine dinucleotide. So yeah, there are two nucleotides. There's one nucleotide, and then here's the other one, dinucleotide, and they are linked in order to create NAD. And here are the two forms of it, NAD plus and NADH. There you go, H, so plus with a plus charge, and then NADH with a neutral charge. That will become important later. So let's jump right into it. Here we have glycolysis. So that involves, again, glucose. That's our main player. And here is glucose drawn right here. So initially, I find it helpful to not worry about the enzymes that can get really confusing really quickly, especially with all the similar names and some names are improper or they could have been done better. So for now, let's just address things conceptually. What's going on? What's happening to the molecule? Forget the forget the enzymes for now. We'll get to that eventually. So the first thing that happens, some enzyme performs chemistry, and here you had, and again, and so a lot of these drawings are identical from the top down to the bottom so that we can see well what is happening, what got added, what got taken off, that kind of thing. So in this case, you can see that a phosphate group was added here within this highlighted orange box. So what this does is that it traps the glucose molecule within the cell. Now that it has this phosphate covalently bonded, this glucose cannot is virtually impossible for it to passively diffuse out of the cell through the plasma membrane. Cool. And again, this is part of what helps with understanding what's going on with the molecule, at least in my case. So for now, we're not covering enzymes. We will get to that soon enough. So then another enzyme performs chemistry and it takes this orange bond that we had talked about, the um, this purple oxygen. It used to be a blue oxygen, which was just a hydroxyl group, but it attacked carbon number one. All right, so that's what made it a glucose. But then an enzyme within the glycolysis pathway, it made that bond um, be put on carbon number two. So now you have this ring that looks like a pentagon. And what happens next is that, okay, so now the molecule is called fructose, specifically fructose 6 phosphate, because it has a phosphate on the 6. Cool. So moving on, uh, another phosphate gets added. Boom, there you go. But now, so now this molecule is called fructose 1,6 phosphate. And that's pretty self-explanatory. There's a 1 phosphate right here on 1 and then 6. Cool. Moving on. So next, essentially what's going to happen is that the molecule we discussed is going to get slashed in this manner. Conceptually, obviously, not, there's more details to the chemistry, but essentially this is what's going to happen. This molecule that we saw up here, again, it's just copied and pasted within this purple box, dark purple box, and I drew these arrows just to show where the split is going to happen. So also the numbering that was present, I've highlighted them in green and in purple so that we can keep track of those numbers a little bit better. We can account for them. They can be held accountable, and we can see what's happening with them. So now that the split took place, now you have 1, 2, and 3 right here. 1, 2, and 3, and it's still in the exact same orientation. I just erased the rest and just highlighted the those three carbons. And over here also, it's still the same. This whatever's drawn here is still the same, drawn the same way. And as a reminder, this purple circle, that's an oxygen. So just a reminder, because this it could be a little confusing, but it does help, I think, ultimately. So um, next up, and in this case, yeah, I'll go ahead and mention one of the enzymes. It's aldolase, and here's the link of where to find this illustration. So essentially, what's happening is that once this severing, this split of the modified glucose molecule, once that split happens, you get this thing and you get this thing. So let's redraw those slightly just to be able to visualize them better. So all of these molecules, all of these atoms, I move them around just to make them a little more linear. And the resulting molecule, one of the resulting molecules from that split is this molecule called GAP, which stands for glyceraldehyde 3-phosphate. And the other one is DAP, which stands for dihydroxyacetone phosphate. Cool. Yeah, so now these are just drawn in a linear fashion, but they're still numbered the same. 1, 2, and 3, and then 4, 5, and 6. All right, so GAP will remain unchanged. Let's leave that alone for now. Let's focus on DAP. What's happening with DAP? Well, here's DAP, again redrawn. It's the exact same thing as above. A plastic surgeon named Tim, his full name is Timothy, he is performing plastic surgery on the DAP molecule. Tim is the name of the enzyme, by the way. It's called triphosphate isomerase. So the reason that DAP is undergoing plastic surgery is because the ultimate goal is for DAP to become identical to GAP. And here I didn't include the mechanism of what happens, how the atoms get rearranged chemically to be exactly like GAP. All you need to know is that eventually DAP, started out like this, but then you go down, 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 and it ends up looking like GAP. So also here GAP, GAP, there's another portion, half of the split molecule already looked like GAP, so that's why GAP is already just um, moved down here or drawn down to here with this blue arrow. So what this implies is that moving forward, whatever is happening to this one GAP molecule is happening to another similar amount of GAP molecules. So anything moving forward is double, as I'm sure you remember. So what happens to GAP is some enzyme comes along and it adds another phosphate. So again, here you go. Over here, as you can see, over here there was no phosphate, but now, boom, now you have a phosphate there. Hence, the name of this molecule is 1,3-biphosphate, biphosphoglycerate. What happens next? Well, this is drawn exactly the same as on top and below. So within this orange box is the next step of what's happening. There exists an ADP just floating around in the cytoplasm. Remember, all this is happening in the cytoplasm. So chemistry happens, and this phosphate group that just got added is it's gonna get transferred to the ADP, which becomes ATP. Boom, and that's how you get this molecule here. It's no longer named 1,3-biphosphoglycerate. Now it's just 3-phosphoglycerate. All right, so what happens next? Well, within this green box, we have that same molecule from the previous step, and again, it's numbered 1, 2, 3. Oh, yeah, that's something important to note. Here, the carbons have been given new numeric values for simplicity's sake and for illustration, better illustration. And as you can see, this one is 3 because it's called glyceraldehyde 3-phosphate. So that was an important note that I forgot to mention. So we had covered this, and now let's go back to where we were. We were here in this green box. So what's going to happen next? Well, the phosphate, instead of being associated with this oxygen on carbon 3, now it's going to get associated with the oxygen on carbon 2. Cool. And again, this oxygen is still written as a purple circle because this way we can keep track of how this oxygen could have been, there's a 50% probability that this oxygen came from the part of the glucose molecule which was which had the oxygen associated with the anomeric carbon. So for now, we'll keep using that purple circle for oxygen. So that's what happened in this step. Now the name of the molecule is no longer 3-phosphoglycerate. Now the molecule is called 2-phosphoglycerate. So that same molecule is drawn here within this orange square or orange rectangle. So what's happening? Well, take a look at, focus on carbon 3. What's happening is that this hydroxyl group on carbon 3 gets taken away. So now the name of the molecule is phosphoenolpyruvate. Cool. And what happens next? Well, that same molecule is drawn, and here you have something's going to happen to the phosphate group. That's why it's highlighted. So once again, there's this ADP molecule just floating around in the cytoplasm, and that phosphate group gets put on the ADP, so it becomes ATP. And now you have just regular pyruvate, which is what you want, so that it can go into the um, next stage of metabolism. And as a reminder, the purple circle here is an oxygen. Cool. So we have pyruvate, and this marks the end of glycolysis. This is the end of glycolysis. So now what's happening is we're getting ready to enter the um, citric acid cycle. So if once you have pyruvate, let's go ahead and quickly redraw pyruvate. In this case, all of the carbons will be brown. They no longer, in this context, they no longer represent the anomeric carbon. In this case, they just represent, they're special because they represent all of the carbons that are that are located on pyruvate, which are going to go into the citric acid cycle. So this pyruvate, and also the purple oxygen right here, that one, that's also not relevant anymore, so I just changed it back to blue. Cool. So there's this molecule called coenzyme A. Here you can see a breakdown of its structure, and what happens is that the pyruvate molecule, so this right here, this experiences decarboxylation. This right here, and there you see decarboxylation took place. That carbon and these two oxygens, CO2, that came off of the pyruvate, and that bond. Now there's this bond between carbon 2 and the sulfur atom of the coenzyme A molecule. Now this whole thing is called acetyl-CoA. What happens next is that you become able to enter the citrus cycle, citrus acid cycle. And here you have some citrus for remembering what's going on. Here, instead of having the whole molecule, acetyl-CoA is just represented by its name. So what happens next is that there's some chemistry that happens, and the acetyl-CoA gets it's no longer involved. There's a handoff of the pyruvate carbons, at least the ones that are left over, number 2 and number 3. Those carbons get put onto a molecule called oxaloacetate. So down here, you can see that I drew the oxaloacetate exactly how it was above, but then I found a way to add the two pyruvate carbons, number 2 and number 3. I added those carbons onto the oxalate molecule, but now this whole thing is called something different. Now it's called citrate, and this is how I happen to draw citrate. So what happens next? Well, here is the citrate molecule again, drawn exactly as above. And what starts happening? Well, let's pay attention to this hydroxyl group, and let's pay attention to this empty space. So there's a transition molecule which is created by enzymes. We'll find out the name of the enzymes later. There's this transition molecule where neither of these carbons have a hydroxyl group, and instead there exists this double bond between these two carbons. But then the enzymes put a hydroxyl group on um, on this other carbon, and essentially what happened is that you see, as you can see here, essentially what happened with this stage of the citric acid cycle is that this hydroxyl group was just moved from this carbon onto the other one, but it's not obviously, as you remember, it's not as simple as plucking it off of one carbon and putting it in another. There was this transition period, this transition molecule, and that's the end result, at least for now. And this molecule is called isocitrate. So what happens next? Well, you have isocitrate, and there's going to be another split of this molecule. So I drew with this red lightning bolt or this red squiggle how this bond was going to get severed, and that's how you achieve this other instance. Once again, you achieve decarboxylation. Decarboxylation. Cool. So this whole other part of the molecule on the other side of the split or the severing, that is a molecule called alpha ketoglutarate. And this is what it looks like. So what happens next? Well, the alpha ketoglutarate glutarate again gets attached to coenzyme A, but here this association isn't called. Oh, and by the way, another decarboxylation takes place. As you can see, I drew things straight up and down. I drew the alpha ketoglutarate on the top and the bottom, but then on the bottom you can see that there is a CO2 molecule that is missing right here. So that means that this was part of decarboxylation, and boom, here you go. Within the blue step, here are those atoms accounted for. Decarboxylation took place. So again, you have this association again of the molecule of interest with coenzyme A, but here the association is not called, what was it called before? In this case, we are not calling it acetyl-CoA. This is a whole different thing. In this case, it is called succinyl-CoA, and this is what it looks like. Cool. So what happens next? Well, you have your succinyl-CoA, and within the matrix of the mitochondria, you have a single phosphate group, and you have a GDP, and there's a lot that goes on. It's kind of complex, and it's not worth running through the details. What you need to know is that what happens is that the coenzyme A, it gets displaced by a phosphate group, and one way or another, this coenzyme A, it just ends up free floating again. So there you go. See, so this is all stuff that happens down from this stage down to this other stage. So the coenzyme A is just dissociated again, ready to interact with the next molecule of interest. And essentially what happened is that this step created a GTP. This single phosphate group became bonded to the GDP, and you get GTP. All right. But what really matters is what happened with the succinyl-CoA. Well, it created this molecule down here, succinate. All right. So what happens next? Well, you have your succinate, and there exists the loss of two hydrogens, and those two hydrogens are now located on FAD, and that's why the molecule gets called FADH2, and we'll address that later. But for now, what happened is that this bond right here used to be a single bond, but now you have this double bond between those carbons. And this is a good time to repeat that these brown carbons are a way of keeping track of which are the two carbons that came in as pyruvate. So we're keeping track of those. So there you are. So now you have this molecule that got created called fumarate. And it always reminded me of fumar, of smoking a Cuban cigar. So for making this a little more memorable, here you have some people enjoying Cuban cigars, including Fidel Castro. Cool. So what happens next? Well, the fumarate then experiences hydration. So this double bond right here now becomes a single bond. Boom. And you have a molecule named malate. And what happens next? Well, oxidation takes place. So this single bond right here, this hydroxyl group, it becomes a carbonyl, and you get that. Well, if we redraw this molecule in a linear manner, just take all of the same atoms and just put them in a line. We redraw them like this. We can ask ourselves the question: who's that molecule? Well, that molecule looks an awful lot like oxaloacetate. And here I have Taurus as a reminder of what we're working with, because his name is Taurus, but you can also think of him as an ox, kind of animal, and that reminds me of oxaloacetate. So what's the deal with this pink arrow? Well, let's find out. The pink arrow is there because we go way back. We go to the beginning, way back, and when you entered right after right as you enter the citric acid cycle, the pyruvate, or whatever was left of the pyruvate, that got added to an oxalate molecule. So how does that tie into the end of the citric acid cycle? Well, let's take into consideration that the oxaloacetate molecule exists here. We're going to follow the pink arrows. We're following the pink arrows. Follow, pink arrows, and boom. So what this is illustrating is the fact that the whole citric acid cycle will take place, and it will regenerate in the oxalate molecule. And here we kept track of the brown carbons, which came in with the pyruvate, but it's irrelevant. At the end of the day, carbon is a carbon within the perspective of the molecule, within the perspective of the cell. It's performing its metabolism actions. So here the brown carbons don't really matter, because what really matters is that we regenerated an oxalate molecule, oxaloacetate molecule. So this oxaloacetate molecule is ready to interact with yet another acetyl-CoA molecule at the beginning of the citric acid cycle. So this got regenerated, and that's why this is relevant and important. All right. So these are screenshots from the Professor Dave Explains playlist. He has this short summary of what's going on with the citric acid cycle. So it begins right here. Remember, it begins right here with acetyl-CoA and oxaloacetate being added or being incorporated. Then you go around, and the cycle regenerates oxaloacetate, as noted here, and it just keeps going in a circle, hence the name cycle. And he also provided the following table in order to provide a summary of what happens at each step. And with these notes, I'll go ahead and expand them in order to include the names of the specific enzymes, and I'll label the steps accordingly so that it can be more comprehensive. But it definitely pays to first just understand things conceptually, and then I'll get back to this. And all of this can be found again in the videos by Professor Dave Explains. Finally, we need to address the electron transport chain, the big payoff. So what's happening is a lot of stuff within the inner mitochondria membrane. And this part, he didn't cover too much in detail, but he did use the important concept, the important key phrase, um, oxidative phosphorylation. And there's a lot of redox reactions that take place back to back to back. That's something I'm rusty in, so I'll definitely be reviewing that and sharing some notes about it in case somebody else would also like a review on that. But for now, essentially what happens is that using all of the NADH and the FADH2, that all those molecules that were generated from the steps of the citric acid cycle, they indirectly create ATP. Indirectly because they don't directly create the ATP, they power these electron pumps, and they push the not the electron, the hydrogen pumps. Those protons get put outside of the matrix into they get put into the mitochondria inner membrane. Sorry, into the whatever's the name of that space. We'll come back to that. And there's that gradient that gets created. So this pump, the ATP synthesis pump, it begins to churn the way that a water wheel would turn, and that's what creates the ATPs, because the gradient, the protons, they're just trying to come back in through this pump according to the gradient that has been created. And that's where you take an ADP and a single phosphate, you associate them with the pump, with the ATP synthesis pump, and you end up with ATP. So again, we'll cover that in detail later. But for now, here you go. Here is that summary. And here's this other summary, that with glycolysis, a glucose will yield 2-pyruvate molecules and 2 ATPs. And the citric acid cycle, it will take the 2-pyruvate molecules and it will yield 2 ATP molecules, 6 NADH molecules, and 2 FADH molecules. And again, that all of those NADH molecules, as seen right here, alongside with the FADH molecules, as seen right here, those will power the proton pumps to create the gradient, and the ultimate net gain of ATP is this amount. The electron transport chain and the oxidative and the process of oxidative phosphorylation, they produce 26 to 28 ATP molecules. Cool. And that is the end.